Welcome to the deep dive.
Flow.
Today we're getting into the weeds, quite literally, with transgenic plants.
Right, what most people just call GMOs.
Exactly. And you've brought sources detailing not just, that they exist, but the actual mechanics, how they're built in the lab.
That's the plan. We're going to break down the engineering pipeline, all 5 steps,
Okay.
and then, crucially, talk about the biosafety side, the ecological questions, especially gene flow. It's sort of a 2-part mission.
Got it. Let's start at the beginning then, the aha moment. When did this actually first work?
It was surprisingly fast, really. The first confirmed transgenic plants popped up almost together in 1983.
83, wow. In what plants?
Things like tobacco, petunia, even sunflower. Several labs were right on the cusp.
And the goal back then wasn't like super crops immediately.
No, not at all. It was purely proof of concept. Can we even do this? Can we get foreign DNA into a plant and make it stick?
Oh, so that explains why the first genes they transferred were often just markers.
Exactly. Like antibiotic resistance genes, kanamycin, methotrexate, things like that.
Simple selection, right? If the plant cells survive the antibiotic.
Bingo. You knew the transfer had worked. Easy readout.
Although for sunflower, they did use a non-bacterial gene early on, right? Phaseolin.
Yes, from common beans. That was a key step too, showing a plant gene could be moved and expressed.
Okay, so that early success sets up the modern pipeline. You mentioned 5 steps.
Yep, roughly.
Right.
Gene isolation, cassette construction, transformation, then regeneration selection, and finally expression analysis.
Let's maybe jump into step 2 first, the expression cassette. That sounds like the core instruction manual.
It really is. That's the package of DNA you're actually delivering.
Before we get to the cassette itself, how did they even get enough of the gene they wanted back then? This is pre-PCR being routine, right?
Right. So it's all about recombinant DNA technology, molecular cloning.
Using bacteria.
Yeah, typically E. coli. You'd insert your gene of interest into a plasmid, get the bacteria to multiply it for you, often interrupting something like the lac operon.
Ah, the blue-white screening thing.
Exactly. A visual way to see if your gene got inserted correctly. White colonies meant success.
Okay, so you've got your gene multiplied. Now you build the cassette. What needs to be in there?
Okay, so you obviously need the gene you actually care about, the one that gives the desired trait.
Right.
You also need that selectable marker we talked about, like antibiotic resistance, to find the transformed cells later.
Makes sense.
Then you need a terminator signal, basically tells the cell's machinery, okay, stop reading here.
Like punctuation for the gene.
Sort of, yeah. And then the absolute key component, the promoter.
The promoter, you called it the ignition switch earlier.
It is. It tells the cell when and how strongly to read the gene. And for most GMOs, you need a very specific type.
Which is?
A strong constitutive promoter. That means it's always on.
Always on. Why?
Well, the goal is usually expression in every tissue, organ, species, and condition. Think about herbicide resistance again.
Ah, okay. You need the whole plant protected, not just, say, the flowers.
Exactly.
Right.
Roots, stems, leaves, everything. So you need that gene firing constantly, everywhere.
Which leads to, well, maybe one of the most surprising facts here. Where does this super promoter come from?
Yeah, this is interesting. The most widely used one, driving expression in maybe 60, even 80% of current GMOs. It's from a virus.
A virus.
The cauliflower mosaic virus, or CAMMV. Specifically, it's 35S promoter.
Why a virus promoter?
Because viruses are masters at hijacking the host cell's machinery. CAMMV evolved this incredibly strong signal to force the plant cell to make lots of virus copies.
So scientists just borrowed it.
Pretty much.
Yeah.
They harnessed that powerful viral on switch to drive their own genes instead. It gives that consistent high-level expression they need. Oh, and importantly, you usually use processed mRNA sequences in the cassette.
Meaning no introns.
Right. You remove the non-coding bits to make sure the plant cell reads it correctly and efficiently.
Okay, cassette built. Powerful viral promoter included. Now, step 3. Getting it into the plant cell. How does that happen?
The workhorse method is, again, borrowing from nature, specifically from a plant disease.
Which one?
Crown gall disease, caused by the soil bacterium Agrobacterium tumefaciens.
How does that work?
Well, Agrobacterium naturally has this huge plasmid called a team plasmid. It contains a chunk of DNA called tDNA.
Okay.
The bacterium actually injects this tDNA into the plant's genome.
Why? What's the goal for the bacteria?
It forces the plant cells to do 2 things. Grow uncontrollably, forming a tumor or gall.
Right, the crown gall.
And produce special compounds called opines, which are basically food for the Agrobacterium. It's a neat, if parasitic system.
So how did scientists turn that into a delivery tool?
The key insight was figuring out that the bacterium only needs specific short sequences at the ends of the tDNA to make the transfer work. They're called the left border, or LB, and right border, RB.
Ah, so anything between those borders gets transferred.
Exactly. So you take out the natural tDNA genes that cause the tumor and make opines. You disarm the plasmid.
Okay.
And you replace it with your custom-built expression cassette, sandwiched between the LB and RB.
So the bacterium still does the injection, but delivers your payload instead of its own disease genes.
Precisely. And typically we use what's called a binary vector system now. The genes needed for the infection process itself, the vir genes, are kept on a separate plasmid from the tDNA cassette. It makes the whole thing safer and easier to manipulate.
It's kind of mind-bending that we're using a natural gene transfer mechanism. It makes you think about the whole unnatural debate differently.
It really does. And, the sources mention this isn't even theoretical. We know natural transgenesis happens.
Like the sweet potato example.
Exactly. The common sweet potato, a staple food crop worldwide, is a natural GMO. It contains bacterial genes, likely transferred via an Agrobacterium-like process ages ago, integrated right into its genome.
So nature kind of beat us to it?
In a way, yes. It certainly adds context to discussions about gene flow and containment, doesn't it?
Definitely. But, okay, Agrobacterium doesn't work for every plant, does it? Some are resistant.
Correct. Or some plants are just really difficult to work with in tissue culture afterwards. That's where you need Plan B.
Which is?
Biolistics, often called the gene gun.
Gene gun. Sounds direct.
It is. You literally coat tiny gold or tungsten particles with your DNA cassette and physically shoot them into the plant cells.
Seriously?
Yep. It's forceful, less precise than Agrobacterium, but for some species, like maybe certain varieties of common bean that are tough to transform or regenerate, it's the only option that works.
Okay, so whether by bacterial infiltration or brute force, you've gotten the DNA into some cells. But that leads to what you call the biggest bottleneck, right? Step 4.
Absolutely. Regeneration and selection. This is done via tissue culture, and it's often the slowest, most painstaking part.
Why is it such a hurdle?
Because you start with just a few transformed cells, maybe in a small piece of leaf or stem tissue in an explant.
Yeah.
You have to coax those cells using very carefully balanced plant hormones and growth media.
To do what?
To first multiply and then differentiate back into all the tissues needed for a whole plant, roots, shoots, leaves, to regenerate a complete T0 plant.
T0 meaning the first generation transgenic plant?
Exactly, the first whole organism you've grown from that transformed tissue.
And the sources say this takes a while.
Oh yeah, weeks, months, sometimes even years, depending on the species and the specific protocol. It requires meticulous optimization and a lot of patience. It's biology, not just engineering.
So you finally get your T0 plant. You're not done yet, though. Step 5 is confirmation.
Right, and this is where the rigorous science meets regulatory expectation. It's not enough just to see it grow in the antibiotic medium.
You need proof at multiple levels.
Definitely. First, DNA analysis PCR, maybe sequencing to confirm the transgene is actually physically present in the plant's genome.
Okay, check the hardware.
Then transcriptome analysis, looking at the mRNA levels to confirm the gene is being transcribed, that it's actually being read.
Check the software is running. And remember, you used processed mRNA, so you need to confirm that specific sequence is active.
Good point. And then crucially, phenotypic expression. Does the plant actually show the trait? Is it resistant to the virus? Does it produce the protein?
See the actual result.
And finally, for any practical application, you need inheritance studies. You have to grow the plant out for several generations, T1, T2, T3 at least, and show that the trait is stable and inherited predictably, usually following Mendelian rules.
So it's not just a one-off fluke in that first TO plant.
Exactly. It has to be a stable, heritable trait.
That stability leads into how we categorize GMOs, right? The different generations based on the traits.
Yeah, the sources break it down into roughly 3 generations.
What's the first generation?
That was all about the farmer, really. Production traits, making farming easier or more efficient.
Like herbicide resistance.
The big one, yeah. Glyphosate resistance, Roundup Ready in soybeans, corn, cotton, and also insect resistance using the beet toxin gene.
But corn, beet, cotton, got it. Anything else major in Gen 1?
Virus resistance was important too, like the papaya ring spot virus resistance that saved the industry in Hawaii.
Okay, so production focused. What about the second generation?
That shifted towards consumer benefits, improving quality.
Examples.
Things like oils with better fatty acid profiles, like healthier canola oil, or tomatoes engineered for longer shelf life, the classic Flair Sauv, though it wasn't a huge commercial success.
And the third generation sounds futuristic.
This is the idea of plants as biofactories, engineering plants to produce valuable non-food products.
Like pharmaceuticals.
Exactly. Industrial enzymes, antibodies, vaccines, other medical proteins, using the plant itself as the production system.
But you mentioned most commercial crops today are still Gen 1.
Overwhelmingly, yes. The herbicide and insect resistance traits still dominate the approved and planted acreage worldwide. The later generations face more hurdles, both technical and regulatory.
Which brings us squarely to risk and biosafety. We've detailed the precision engineering. Now let's talk about proving it's safe.
Right. And the sources distinguish between food safety and ecological safety.
How so?
Food safety, while complex, is somewhat more contained. You're testing for things like allergenicity, toxicity, nutritional changes. It involves lab tests, animal feeding studies, similar in principle to testing a new medicine.
Okay, manageable. What about the ecological side?
That's much harder. The big challenge is assessing potential impacts on non-target organisms.
Like the monarch butterfly issue.
Exactly. The famous study later questioned and context put around it looked at monarch larvae fed high doses of boot pollen in a lab setting.
And found negative effects.
At those high lab doses, yes. Even though it was unlikely larvae would encounter that concentration in the wild, it highlighted the need to consider these off-target effects.
So it forced more rigorous testing.
It absolutely did. Proving safety across a whole ecosystem is a vastly more complex task than proving safety for human consumption.
And arguably the biggest ecological concern discussed is gene flow, right?
Yes, defined as the transfer and stable incorporation of that engineered transgene into the gene pool of another population.
That could be the same species like conventional corn or a different related species.
Correct. Either way, if it's unintended, it can cause problems.
What kind of problem?
Several. One is genetic erosion. If transgenes flow into traditional varieties, the local land races, you could lose valuable genetic diversity farmers have cultivated for centuries.
Oh, okay.
Another is reducing genetic purity, which matters for markets that demand non-GM products. Contamination is a real economic issue.
And the third?
Increased weediness. If a transgene, like herbicide resistance, transfers to a related wild species, that weed could become much harder to control.
Makes sense. How do scientists categorize the risk here? You mentioned gene pools.
Yeah, they think about it in terms of how easily the crop can interbreed with wild relatives. The primary gene pool means easy naturalization can happen.
Like between different varieties of the same species.
Exactly. The secondary gene pool means crossing can happen, but it's less frequent, maybe needs some help. The tertiary pool means crossing is very difficult, basically requiring extensive human intervention in the lab.
And how does the gene actually flow? What are the routes?
Two main ways: seeds and pollen.
Seeds seem obvious if they spill during transport or get mixed in storage.
Right. Or even just volunteer plants sprouting the next season from leftover seeds. Pollen flow is more about biology, wind pollination, insect pollinators.
And distance matters there, presumably.
Hugely. Distance, wind patterns, the type of pollinator, even physical barriers like hedgerows can all affect how far pollen travels and potentially fertilizes a non-GM plant.
So, given these risks, what are the strategies for containment? How do you ensure coexistence?
There are genetic strategies and cultural strategies.
Genetic ones?
Things like putting the transgene in the chloroplast or mitochondrial DNA instead of the nuclear DNA. These organelles are usually inherited maternally through the egg cell, not pollen.
Clever. No pollen transfer.
Or older ideas like Terminator technology, engineering the plants to produce sterile seeds, though that was highly controversial and isn't really used commercially.
Okay. And cultural strategies?
These are management practices, setting up mandatory isolation distances, geographical exclusion zones, planting buffer rows of non-GM crops around the GM field to trap pollen.
Like a biological fence.
Sort of. Also, strict protocols for cleaning equipment, managing storage, and importantly, actively removing any volunteer GM plants that pop up where they shouldn't.
It sounds like an immense amount of effort goes into containment after the plant is made.
It has to. The regulatory requirements for demonstrating biosafety are extensive.
Which leads to something you touched on earlier, the innovation barrier.
Exactly. All this testing, proving safety for food, feed, non-target organisms, managing gene flow, environmental impact assessments, takes years and costs millions upon millions of dollars.
Per product.
Per transgenic event, essentially. Each specific insertion needs full validation.
So who can afford that?
Realistically, mostly large agricultural corporations. It creates the huge financial barrier for smaller companies. university labs, or nonprofits who might have genuinely beneficial innovations but simply can't fund the deregulation process.
So the complexity of ensuring safety inadvertently limits who can bring these products to market.
That's a fair assessment, yes. The cost of regulation is a major factor shaping the landscape of agricultural biotech.
Despite that, adoption is massive globally, isn't it?
It is. The U.S. plants the largest area, over a third of the global total. Brazil is the top developing country adopter.
And it's not just big industrial farms.
No, the data shows millions of smallholder farmers benefit too. In 2019, something like 17 million small, resource-poor farmers were growing biotech crops.
Okay, so we've gone from gene isolation to global adoption and regulatory hurdles. Let's wrap up with that final thought you wanted to leave us with.
Right. So we've talked about the immense effort for containment driven by safety and regulation. Absolutely necessary. But we also acknowledge that transgenesis happens naturally. The sweet potato is proof. Genes jump between species over evolutionary time.
Okay.
So the provocative question for you, the listener, is this. Given that natural gene flow occurs, does our current, extremely rigorous focus on containment, essential as it is for managing risk, now paradoxically slow down our ability to adapt crops? Could it prevent us from deploying potentially vital traits fast enough to meet huge future challenges like climate change or food security?
Wow, that's, yeah, pitting necessary caution against potential future need. A really tough balance.
It is. There's no easy answer there.
Definitely something to chew on. This has been a fascinating deep dive into the mechanics and implications of transgenic plants. Thanks for breaking it down.
My pleasure. It's complex stuff.